This conference will now be recorded.
So, yeah, thank you for the invitation from the RFMF for giving me the opportunity to present my postdoctoral research, which is about combining innovative NMR method to improve environmental contaminant characterization using Metabolomics. So, with all the audience that we have today, you probably all know the challenges that are linked to NMR metabolomics. The main challenge is very low sensitivity that NMR presents, but there is also several other small challenges, such as the fact that we are looking at a large range of concentration, and that will impact the digitalization of small peaks, which brings the fact that it's very important to suppress the higher signal, which is most of the time the solvent signal, but still by maintaining a good robustness and repeatability in order to maintain intra and inter-laboratory comparison, and maintaining quantitative information as well, because it's one of the assets of NMR metabolomics. But we are dealing with all that, and dealing also with the fact that because we are looking in metabolomics at complex mixtures, we are looking at a great variety of molecular structure, which give us spectra very crowded with a lot of signal overlap. So there is a GoToMeeting camera that is coming on my screen every time. Okay, should be good now. So there is a lot of method development that is being done to overcome the current NMR metabolomics limitation, and a lot of them have been done in the Sesame laboratory, especially to make NMR more sensitive with hyperpolarization, NMR more accessible also with benchtop NMR, better integrated in order to combine the asset of NMR with the assets of mass spectrometry. But method development that is being done and that will be of interest today are made on 2D NMR to have a better result spectra. But the problem with 2D NMR is that we need to have two temporal dimension. Obviously, we don't have two temporal dimension, and in this case, that means that we need to have several acquisition, one after the other, on the same sample, which made the acquisition time obviously very long and not compatible with the high throughput criteria of NMR metabolomics. So there are a lot of different ways to make 2D NMR more rapid and make it actually compatible with the high throughput criteria. And those time-saving strategies, so I'm going to present you three of them today that I base my acquisition on. There is ultra-fast, where basically the sample is cut in different slices, and there is a simultaneous acquisition of the NMR spectra on the different slices. So this really improves the rapidity of the acquisition. There is also the non-uniform sampling, so commonly called NUS, where only some of the points of the spectra are going to be acquired, and the rest is going to be reconstructed by an algorithm. And it's the person who is acquiring the data who is going to decide of the proportion of the data that should be real and the proportion that should be reconstructed by the algorithm. And spectral aliasing, it's basically just as if you were folding a paper in two, and so the signal that are in the part that is fold on the other are going to be reported on the first one. So it's going to actually cut in half the acquisition time. And with these different time strategies that offer a continuum of fast 2D NMR method that represents a compromise between speed, sensitivity, and resolution. And it's offer all of them together offer still a better resolution than 1D NMR and allow to overcome the overlap problem that we have with 1D NMR. And so that's what we decided to apply in my postdoctoral project, where we combine those three different methods that I've presented you just in the slide before. But because we wanted to, those acquisitions have already been applied to metabolomics, but the complementarity or the redundance have never been explored. And the complementarity and redundance with the second keystone of metabolomics, so mass spectrometry, have never been explored either. So if we want to propose to the metabolomics community a new method, we need also to prove that there is an interest into it. And so that's why we decided to apply those fast 2D NMR method with 1D usual metabolomics acquisition, plus on the same sample, and still on the same sample, apply also 9 different IHRMS datasets. And to explore the complementarity, but also put those different analytical methods in competition, we decided to apply a multi-block data integration with in collaboration with the University of Geneva. So I'm going to present you quickly first the study design, the sample preparation, and the data acquisition. So because the mass spectrometry part were in collaboration with the Laberca, we had access to serum sample coming from mini pigs that were exposed or not to bisphenol A. And following a simple blind Dyer sample preparation, we were able to collect the aqueous phase to look at the metabolomics part, but also the organic phase, and to perform a lipidomics on the organic phase. And so on those two phases, we acquire 4 different NMR datasets, the 1D classical dataset in 15 minutes for each sample, the 2D ultrafast COSY in 15 minutes, the 2D non-uniform sampling TOXI in 1 hour, and the 2D per shift HSQC in 2 hours, which represents 4 hours of acquisition per samples. And so what we wanted to do then with those different datasets, it was to obviously in the ultimate goal to put them together to explore, to put them in competition through multi-block data integration. But we first wanted to look also at how each of the individual data sets were performing. So what we've done is that we perform an OPLS-DA, so supervised statistical model on each of the data sets. And we then look at I don't know if you can see, you should be able to see my pointer now. And we also then combine each of the 4 different acquisitions for the metabolomics, just to compare the different NMR method. We've done it also for the lipidomics, and we finally combine everything in a global consensus OPLS DA to look also at the advantages of combining metabolomics and lipidomics. And so I should also precise that we've done a variable selection before to perform all the different statistical model in order to focus ourselves only on the variable the most informative. And so we choose to apply the consensus OPLS-DA for the multi-block integration because it's a package that has been developed and it's available on MATLAB and has been developed by Julia Bocca from the University of Geneva. Okay. So now going on the results, so I've presented here the performance indicator of the different models, so the individual one and the combined one for the metabolomics and the lipidomics, with a quick reminder that the R square Y is representing the goodness of the fits, while the Q square Y is presenting the goodness of the prediction, and the R square X is rather looking at the which proportion of the variation of the variability is captured by the model. And even if it's an arbitrary value in metabolomics, we cross often this Q square Y value of 0.4, and that above it's a model that we can have confidence in, and if it's below 0.4, it's not a model that performed in terms of prediction quite well. And if we look at this table here, we can see that only the 1D metabo and the 2D ultra-fast COSY for the metabolomic part as well are above 0.4. But if we look at only the metabolomics, so this is the score plot, the OPLS score plot that is presented for the 1D dataset, we can see that actually the proportion that of variability between the sample that is explained by this model is quite low. If we look at the lipidomic part, we can see that the Q square are quite, especially for the 2D, are quite low, even negative. So that means we cannot really have confidence in those models that are not managing to separate and see any difference between the two groups of samples. If we look now at the integration of the 4 different NMR datasets for the metabolomic part, we can see that the Q square and the R square Y are quite satisfactory and didn't change that much for the goodness of the prediction. But we still increase the proportion of the variability between the sample that we managed to capture with this model. And if we look at the different block that has been used for this model and how they participate to this discrimination between the sample, we can see that it's actually the 3 2D data sets that are participating and capturing the most variability between the samples. And my explanation behind that is that because the signals are much more resolved, it's actually there is the buckets and the integrals are of better quality, which means that the statistics also are of better quality as well. So this is very encouraging for the use of 2D NMR in metabolomics. If we look now at the lipidomics dataset, so all combined, we managed to increase also the Q square and the R square, still by capturing a large part of the variability compared to metabolomic particularly. And once again, it's one of the 2D NMR datasets that is managing to capturing most of the variability between the samples. But where it's becoming even more interesting, it's when we are combining the metabolomics dataset with the 4 lipidomics datasets, where this is where we manage to have the highest performance indicator, and still by maintaining a good proportion of variation that is being captured. And this is because obviously, by combining metabolomics and lipidomics, we are covering a larger part of the metabolome, and that means we are able to understand a larger part of the changes, of the biological changes that is happening because of the bisphenol A exposition, which help us to have a more confidence in our model. So what will be next in this project? Obviously, the biomarker identification following the NMR multi-block integration, but also GC-HRMS and LC-HRMS have been acquired for the LC in Metabo and Lipido, and in both phase, reverse phase and helic and post-neg each time. So the processing of all those data sets is undergoing, and obviously the ultimate goal will be to combine everything and to see if those methods are complementary or sometimes redundant, and help us to choose which method we should select and we should go for once we are starting a new project. So now the acknowledgements. I would like to thank obviously all my colleagues that helped me on this project at the Sésame Institute, but also all the friends from the Laberca, and a special thanks to Julien Bocard from the University of Geneva for all his help on the stats and the multi-block integration. And thank you to you for your attention, and I will be happy to.
Thank you, Marine, for this presentation. We already have one question in the chat. The question is, what about the 2D NMR pre-treatment to extract the data according to the fact that it could be more complicated than 1D workflow?
It's a very good question from Pascal. Yes, it's quite complicated, especially because some of the data are quite huge, and the main problem that I'm facing now is that when you are processing your 2D NMR, either you have the size of your data are not that important, and the processing is actually quite easy. I will come back to that later. But when the size of your data are quite huge, the software that we have currently to process 2D NMR don't support the size of the data that we have, especially for the toxic here. So it's okay when we are looking at one spectra at the time, but when we are looking at them in a batch mode, it's actually impossible, and every software will crash. But when the size of the data set is not that important, it's actually quite easy. So we had an engineer here at Sésame Institute to develop the graphical user interface on MATLAB to automatically integrate all the different signals that are on the spectra. And that's, in this case, it's very easy, and there is way also on topspin before to correct the baseline or phase all the different spectra quite quickly. And there is we are not doing alignments. It's not that needed on this study here, but there is only 80 samples, so maybe that's why it's not that important. But yeah, it's just linked to the size of the data that we are having problem. Otherwise, there is solution to process the 2D.
Okay, thank you, Marine. Maybe I could ask myself my question. In terms of redundance of the metabolites, when you make the multi-block analysis, how do you deal with that redundance of that metabolites? For instance, so if you have, okay.
We are not dealing with redundance at all now.
So you let the information in each block.
Exactly. Okay. We let all the same information in each block because there is two ways to see the redundance. There is the fact that redundance it's actually a drawback, and it could introduce a bias in the statistical analysis. But if we have the same metabolite that is coming from a statistical point of view, if we have the same metabolite that is from different block that is coming out as significant, it's can also be seen as a validation, and you see if this metabolite is actually coming out as significant in all the block where it appears, it's actually reassuring, that because that means it's actually very significant. But still, we want to look at the redundance after that. But for that, we need to be able to know where the redundance is, and for that we need to identify all the different signals that we have in all the different data sets, and because there is 17 data sets in all between NMR and mass, it's gonna take a little while, but it's a PhD project that we have for next year.
Okay, thank you so much.
Thank you, Marine. I think we still have time for one last question, and it's do you think that using labeled BPA can give more precise results?
To perform flux analysis afterwards, or
I think
not sure
this is maybe.
But yes, maybe. That's to be totally honest, this is not a question that we asked ourselves. We took those samples because they were already available, and we could have taken any samples to test the 2D NMR, still with a tox preclinical and clinical application. That's our interest. But yeah, we were not in the team of the to plan the study design, so I don't know. Maybe it could help.
Okay. Thank you again, Marine, for this presentation. And as Pascal said in the chat, we are all looking forward for future development in this field.
Thank you very much.
And now we are moving to our next presentation that will be given by Ariana Cirillo, who is currently finishing her PhD in the University of Liège under the supervision of Pascal Detulio. During her PhD project, she was mainly interested in identifying a molecular signature of delayed graft function of kidney allograft using NMR metabolomics. So Ariana, the floor is yours.
Thank you, Gina, for your introduction. Are you able to see the full screen?
Yes.
Okay, thank you. Thank you to the RFMF that give me the possibility to present my project that is based on the study of untargeted NMR-based metabolomic for the identification of metabolomic signature of a delayed graft function. Kidney transplantation represents the best treatment for a patient with end-stage renal disease. And in particular, each year worldwide, 90,000 kidney transplantations are performed, but still the outcome depends on the graft quality. Defined guidelines have been listed in way to define as a series of standard criteria donor, which may guarantee the graft quality by including as donor only people younger than 50 years old and which suffered from brain death and that are known as a dead brain donor. However, the increasing gap between the demand and the supply for the kidney transplantation has expanded these standard criteria donor for generate the extended criteria donor, which additionally add to the dead brain donor donors for which the death is confirmed via cardiorespiratory criteria, and also known as a dead circulatory donor. The problem with the kidney coming from these suboptimal donors is that they are exposed to a prolonged ischemic event, which generate in a patient a condition named delayed graft function in the post-transplantation period. So when we look at the transplantation timeline, we can directly spot the difference between the dead brain donor and the dead circulatory donor. In fact, if we look at the dead brain donor, in this kind of donor, actually the blood flow never stops between the death event and the start of the cold ischemic period, compared to dead circulatory donor, in which between the circulatory unrest and the start of the cold ischemia time, there is a gap of time that is named warm ischemia time, that is directly correlated with the possibility of patient to develop a condition of delayed graft function in post-transplantation period. That's why in our study we focus on the analysis of the perfusate that represent the cold storage solution that is injected in the kidney and which represent a real mix of all the metabolites that are present in the kidney just before the transplantation. So in this study we focus actually on two main questions. The first one is the possibility to have a panel of biomarkers predicting the DGF condition. And as second question, the possibility to define and to identify a difference in the metabolome between BD and DCD donors. That's why in this study, as we see in the context of kidney transplantation, there is a real need for identify a tool able to assess the graft quality after the kidney procurement and storage and just before its transplantation. And for this reason, in our study, we propose a solution, the application of an untargeted NMR-based metabolomic approach, in which to detect all the metabolites that are present in the perfusate, with the aim to define a panel of biomarkers predicting the occurrence of DGF condition post-transplantation, and to explore also all the biochemical events that are occurring between the death and the hypothermic storage of the kidney. In this study, as first we focus on the difference in the metabolome between the two kinds of donor. And for this, for our study, we use an experimental model and a human study. In particular, for the experimental model, perfusate sample coming from rat were classified on the basis of the donor type, DBD and DCD. And the same was done for the human sample. After NMR analysis and the identification and quantification of metabolite through genomics, we were able to perform our univariate and multivariate statistical analysis. So as we can see, as first result, here we have in comparison the experimental model and the human study, from which we can see that there is a clear separation between the DBD and DCD donor that is more accentuated in the experimental model. For go deeper, we make and we perform an univariate statistical analysis that allowed us to identify all the metabolites that were related to this difference, to this different type of donor. And in particular, we were able to see that there were some metabolites that were common to both models and that were evolving with the same trend. So at this moment, we perform a biochemical pathway analysis that allowed us to see again that the same biochemical pathway were actually involved in both experimental and human study, by confirming the fact that actually the biochemical events that are impacting the donor type are the same. So as a take-home message from this first part of the study, we can say that both the experimental and human study allowed us to characterize the difference in the metabolome based on the donor type. And also this difference in the metabolome need to be further investigated in way to improve our knowledge regarding the biochemical events that are occurring between the death and the hypothermic storage of the kidney. At this point, we decide to focus our attention on the DGF status post-transplantation. In particular, for do that, for our study, we use perfusate sample coming from a human cohort, which was classified based on the post-graft status, so on the possibility of patient to develop a delayed graft function post-transplantation or unhealthy condition. Two additional analyses were done in this context by dividing our human cohort at first on the basis of the donor type, so DBD and or DCD, and after by classifying them on the basis of the pathological status post-transplantation. So in the next slide, I will show you only the result concerning the general analysis and the result concerning the analysis made on DBD donor. Since, as we can see, for DCD donor, the number of each patient for group are really reduced for a robust and reproducible result. So as first, for the general analysis, so made on both type of donor, we use as discriminant factor the DGF outcome post-transplantation. And as we can see from the PLSDA, the performance is not good, but we were still able to identify some metabolites that were relevant for the separation between the healthy and non-healthy status. At this point, we decide to take only the donor type, so also only the dead brain donor type, and on this group made the separation based on the DGF status. And as we can see, in this case, the performance of the model is really increased, passing by a Q-square of 0.37 to a Q-square of 0.74 in this model. And also in this case, we were able to identify the significant metabolites. What we can highlight from this result is that it's really important to take in consideration the donor type before to look at the pathological outcome post-transplantation. So for go further in this analysis, we perform a prediction model. So as first, we generate a multivariate log curve by which we were able to see that the performance, the prediction performance of the model was good, with an AUC of 0.92 by taking in consideration 36 variables, and also a good confusion matrix was obtained. At this point, the metabolites that were found to be significant in the univariate analysis were used for generate a metabolic pathway analysis by highlighting all the metabolic pathway that were actually impacted by the DGF condition post-transplantation. So as a take-home message from this part, we saw that the impact of the donor type is something that has to be taken in account if we want to make other analysis and look at the DGF condition. Additionally, this NMR-based metabolomic approach may predict the occurrence of DGF condition post-transplantation, and in particularly the identification of this biomarker could represent a real tool for help clinician in clinical practice. So as a general conclusion, through this study, we were able to improve our knowledge about the biochemical pathway relate to the hypothermic storage, relate to the donor type. And we were able to identify a set of biomarkers linked to the DGF condition that could help a clinician in the management of post-transplanted patients. And as a perspective, we hope that we will be able to set up a validation cohort in way to confirm the metabolic profile of both donor type and DGF outcome. In conclusion, I would like to thank all the people that participated to this project, all the components of the Metabolomics group of the University of Liège, the RFMF for giving me the possibility to present my work today, and for sure you for your attention.
Thanks, Ariana, for your great presentation. Do we have questions in the chat? Not yet, so I'll start with my first question. So it concerns the first part when you compare like DBD and DCD on your animal and human study. I was wondering if you have any biological hypothesis of why such amino acids would be increasing. I can't remember if it was a DCD or DBD condition, but maybe could it be related to, I don't know, protein degradation, or it might be a silly question, but
yeah, no, totally. It's a really interesting question. Thank you, Loic. And actually, we made some literature, and there are some reports with already make this kind of differentiation between DBD and DCD donors. But in the case of liver transplantation, and also in this case, actually, they were seeing an increase of certain amino acids that are related to the metabolic degradation that is happening actually in the organ in general during the warm ischemic time or the cold ischemic time. And the level of change of this amino acid actually it's changed on the basis of the technique that is used or the preservation solution used. So there are a lot of different parameters that has to be taken in account for look at the variation of the amino acid. But is really something interesting at which we want really to better investigate for understand which are all the biochemical process and why these biochemical pathways are actually activated in one case or not in another.
Great, thanks. Yeah, I think we are a bit short on schedule, so if anyone have one last question, we can. Otherwise, we're gonna have to move on to the next presentation. Okay, so let's move on. Oh no, there is one last question in the chat. I was wondering if there are any plans to translate your results to a larger scale. So maybe the validation cohort, I suppose, or something.
Yeah, for sure. This is really the aim of our perspective is really to have a validation cohort for be able really to confirm what we found or not. But I mean, the main aim is really to translate actually all our research in a clinical practice for really generate a tool that could help really clinician for the follow-up post-transplantation, and that is really a tricky period and a tricky moment for both patient and clinician.
Okay, so there is a very last question. So if I have understood correctly, the patients included in the study are close to 50 years old. How can we be sure that their pathological profile does not influence their metabolic profile and therefore the metabolites identified?
Yeah, so Yeah, it's a really good question. And actually, I didn't mention it, but in the standard donor criteria, the donors has to be younger than 50 years old, but also need to respect some pathological criteria, such as never had history of hypertension or some other pathological condition that may affect the quality of the kidney and the organ. So actually, it's really important to have an historical of all the pathology and all the health status of the patient.
Thanks, Ariana, again. So now we have to move on to Jessica's presentation.
Thank you.
So Jessica, I think you can start to share your screen if you have the right. Otherwise, let me know. So Jessica has recently defended her PhD, which was carried out at the CEA in Saclay, under the supervision of Florence Castelli and François Fenaille. And her main focus during her PhD was to study the metabolic rewiring of dendritic cells upon TLR activation. She used MS-based metabolomics, so it's going to be good to hear a bit of mass spectrometry after all this NMR talk. So can you share your screen? Now you should be able to share it.
Yeah, sorry, Loic. I'm here. But I can share my my screen. Okay, thank you.
Now it should be fine, yeah.
Yeah. I will not put my camera because the Wi-Fi is not really good. So to be sure.
Okay, we can see your screen.
Yeah, thank
Perfect.
you. So good afternoon, everyone, and thank you for the invitation. So today I will present you the investigation by metabolomic analysis of the metabolic rewiring of dendritic cell upon activation. This project was part of my PhD project that I conducted at CS Saclay under the supervision of Florence Castelli and François Fenaille. But first, let's start with some basic knowledge of immunology. So all living organisms have immune system that can be activated by harmful stimulus. This stimulus will induce a complex biological process called inflammation. It will be the innate immune response that will be first activated, and it will induce a rapid and non-specific response. During this response, different cells will be actors, and here we are focusing on macrophages and dendritic cells due to their great ability to phagocyte and neutralize microorganisms rapidly, but also because they have the capacity to activate adaptive response that is much faster and specific to the inflammation. The activation of the cell is due to the recognition of pathogenes associated molecular pattern, an identity card of microorganism by receptor at the surface of the cells as toll-like receptor. This recognition will induce a lot of change, first morphologically, then they will increase the secretion of cytokine, but also they will induce a metabolic change. Cellular metabolism was studied with a targeted analysis, and we call immunometabolism field. It's appeared in 1915, and they succeed to describe some alternative pathway like glycolysis, the PPP, or the Krebs cycle, but also some intermediate of the metabolism with key roles in the immune response, such as pro-inflammatory with succinate, citrate, or itaconate. But from this simple picture, we can clearly see that the scope of investigation is limited. That's why immunometabolomics emerged, wanted to analyze all the metabolome of the cells, and that will be done with the use of data-driven analysis. In this project, we will use high-resolution mass spectrometry coupled with a chromatography liquid, and we will use 2 different column and ionization mode to better coverage of all the metabolome. But can we believe that we are the first to think to use this methodology? And if we look at the range of the rate of publication on this field, focusing on macrophages and dendritic cells, we can clearly see that the field was known a great emerged in 2015 and grow since now. And we can also observe that the large majority of the publication is about macrophages, here in red. And this can be due to the better and the facility of manipulation and culture of these cells, but also because there are a lot of cell lines on the market. So it's in this context of lack of metabolic analysis on dendritic cell that this work fits in, and we wanted to investigate the metabolic reprogramming of these cells with untargeted analysis. But first, the first step of this project was to set up an optimized metabolic protocol by questioning the time of exposure and also the protocol used for the collection of the sample. Quickly, the workflow will begin with the obtention of monocyte-derivative dendritic cells from buffy coat of healthy donors. The cell will be then activated by LPS, that mimicking the membrane of gram-negative bacteria. And after the harvesting step, we can conduct a metabolic profile by using the LCHMS and also MS2 identification with in-house database. First, we will identify the cell of interest by counting the cell attached at the flask separately from those present in the supernatant. This during 48 hours of kinetic. A significant increase in blue of adherent cell was observed, beginning to 2 hours and no degrees at 48 hours. That means that activation range is about around 24 hours. To be sure that the adherent cell are well activated, we were analysis marker linked to activation state. Here, I will present you the CD83, and we can observe that the adherent cells express highly the CD84 during time. That means that the adherent cells are well activated by the LPS. However, cell adherence means that important harvesting methods have to be applied. For metabolomic study, it is important that the collection step is rapid and does not induce any bias for the rest of the study. In addition, the method must be applicable in 2 scenarios. After differentiation, so for non-adherent cells, and after activation for adherent cells. The most common protocol is the use of trypsin. For metabolomic analysis, this step will be followed by organic solvent extraction. The second method, which is becoming increasingly popular, is the one-pod method, which which will simultaneously extract the intracellular metabolite, but also stop the metabolism by applying a cold organic solvent directly into the flask. The disadvantage of this method is that no intact cell material is available for further analysis. By looking at the literature, we saw that in 2009, Tang was described that the use of trypsin can cause damage to the membrane, leads to leakage of metabolites, and induce cellular stress. So by applying these two methods and looking at the associated metabolic profile, we can quickly observe via principal component analysis that the two profiles are quite distinct. Here in red, we have the sample harvested by trypsin, and here by organic solvent. Doing a further analysis, we also observe that more than 80% of the metabolites detected were decreasing in presence of trypsin, validating the observation of Tang and confirming that the one-pot method is the suitable protocol for immunometabolomics. The first experiment that we conducted was a kinetic over 5 time points on 12 buffy coats from healthy donors. During a single run, we were able to observe over than 3,000 robust variables, thanks to use of these two chromatographic analyses, and more than 200 metabolites at a high level of confidence, thanks to use of MS2 identification with in-house database. By applying an heat map, we were able to observe the evolution of all the metabolites over time, and 4 main groups was emerged, those decreasing over time, the group with increase only after 16 hours of activation, those that increase after 2 hours but return to the initial state, and the majority of the metabolites there was the first kinetic on dendritic cell and the first metabolic profile that was conducted on these cells. From this first observation over time, we were able to propose a possible immunoregulatory circuit. So by observing at the same time the production of polyamine with putrescine spermidine, despite the degradation of ornithine and arginine, and also the degradation of tryptophan for the production of quinoline and quinoinic acid. Tryptophan into quinoline could be promoted by the activation of RHR receptor, already described in the literature. And also, we found in the literature that the production of quinoline can have a positive influence in RHR activity. Moreover, RHR activation to polyamine, and to go furthermore, we observe in the literature that the production of spermidine can have a positive influence in IDO. So with our data and published result seems to suggest a wider retroactive loop involving all this metabolism, but this first observation have to be confined at the mechanical level. But based on this first result, we were also questioning the specificity of metabolic change of dendritic cell under different activation. So here we compare the signature of 24-hour bacterial activation with viral activation mimicked by poly-IC. The PCA study of the metabolic profile show only a good separation between unactivated cell, here in red, and activated cells. By two different metabolic pathway caught once again our attention, and it was tryptophan and urea cycle. What we observed that the data seems to indicate a similar trend to the formation and production of quinoline and quinolinic acid, but with more intense change in case of viral here in red. At contrary, only poly-IC seems to induce a significantly production, here still in red, of a citrulline. First, those time effects have to be implemented to understand this important production of these two tryptophan intermediates. And also mechanistic study have to be implemented to highlight the possible use of INOS enzyme during poly-IC activation, meaning that maybe under this viral activation, nitric oxide production is favorable. So further investigation have to be done on this really first primary result. To conclude, metabolic reprogramming of dendritic cells was successfully recalled by thanks to LC-HMS method, but also thanks to the setup of switchable protocol for adherent and non-adherent cells. We were able to compare immunometabolome under different exposition. We have managed also to have more than 200 metabolites robustly monitored, affected over all the time, and we highlight fine distinction of metabolic profile. This methodology is also possible to apply to other immune cells. But there are still challenging areas. It seems important to work on significant unknown data because in our project it represents more than 20% of significantly altered, and also to integrate multi-omic, multi-block analysis to really combine metabolomic, lipidomic, and cytokine analysis. This project would not have been possible without the help of Florence Castelli, François Fenat, and Christophe Junot, as well as all the other colleagues at LIMS, Elideza, Léry, and CIMOS. I would like to thank the RFMS and RFMS Junior too for the invitation and for giving me the opportunity to present this work. And finally, I would like to thank you for your attention.
Thank you very much, Jessica. It was a pleasure to hear you. So I don't see some question in the chat, but I have a question for you. Do you think to confirm the implication of the citrulline consequent to viral infection with other virus like influenza virus, or do you plan to do that experiments or not?
So first, the idea was to use product that mimic the activation of the TLR. But of course, we can think to use real virus, but you will maybe need to implement biological protocol and the safety protocol first. Okay. But we want to confirm this first result with stimulating project.
Okay, ligand. Okay. Okay, so I think we are a little bit late. So if there is no more question, we can move on our next speaker. So thank you again, Jessica. It was a pleasure. So our next speaker is David Wishart. And it's my pleasure to welcome David today. And David is a professor in the Department of Biological Science and Computer Science at the University of Alberta, where he led the Human Metabolome Project, a multi-university project that catalogued all known metabolites in human tissues and biological fluids. And this project led to the creation of the database that you are all familiar with, that is Human Metabolome Database, HMDB. And today, David will share with us his view on how we can move on from metabolomics to more multi-omics approaches. And so it's a real pleasure to welcome you, David, and I hope that you have this vision and you can share with us, with all the community of the RFMF, this vision. So you are here, and I think you can start your presentation.
Yes, I don't know if you can see my screen right now. Is that okay?
Yes, it's okay.
Great, and the audio is also okay. Thank you very much, Audrey and Lorik and others for inviting me to present today. It's a pleasure to be here, and I'm hoping that what I can do will at least inspire some thought and maybe some discussions for a later part of this seminar. So I'm talking about how to move metabolomics towards multi-omics, and this is really based on a review that I wrote about a year ago, which was called Metabolomics and the Multi-Omics View of Cancer. And it was sort of partly an opinion piece, but also one that I think had some fairly useful insights and data about why and how we need to rethink how we approach and think about cancer and how we approach and think about other kinds of diseases. And as part of that review, we have this illustration which showed these connections that people have traditionally and more recently viewed cancer with. On the left-hand side, we have this picture of how the genome leads to the transcriptome, which leads to the proteome and the metabolome. And these are sort of endogenous activities that happen. And cancer has traditionally been viewed as something more of a genomic disease. And in fact, the very first important genes identified that cause cancer are called oncogenes, and that's still where our major focus is. Of course, oncogenes are transcribed to produce oncogene transcripts. Those can then produce sort of mutated proteins that are called oncoproteins. More recently, metabolomics has entered into the field, and there are now metabolites, which are called oncometabolites, that also seem to contribute to cancer. So we have terms like oncoproteins, oncogenes, oncometabolites. We have metabolomics and proteomics and genomics, all being part of the puzzle of understanding cancer. But on the other hand, people also now realize that cancer isn't purely a genetic disorder. It's not internal. It's actually caused by a number of other external parameters. What you eat, what you're exposed to, radiation, viruses, and bacteria. And these on the right side are part of what we call those the exposure or external or exogenous contributions to disease. So the chemicals or the radiation or other things that are external to us are now called the exposome, and then the microbes and viruses, which are also known to cause about 20% of cancers. Those are oncoviruses and oncobacteria, and that's part of the microbiome. So what we can think of now is that cancer isn't just a genetic disease, it's a multi-omic disease. And in fact, we can extend this to probably all other diseases and pathologies that we think about, or just even conditions that they are or contribute or see contributions from the genome, transcriptome, proteome, and metabolome, but they're also often due to exposures or pathogens or microbes. And thinking of disease as only one silo, of only one omics, of only one form is probably too naive. And even though most of us here are doing metabolomics, we also have to think of other contributors to the conditions that we're looking at. Now, one of the problems with analyzing omics data is that we tend to work in silos. metabolomics, people use metabolomics data and metabolomics software and publish in metabolomics journals. People doing genomics use DNA sequencing, use specialized DNA analysis software and publish in genomics journals. The same is true with proteomics and exposomics. And none of them really are talking to each other. The question I'm asking is, does it really have to be this way? Do we have to continue living in these silos and doing our research isolated from other fields of omics science and not considering the contributions that, say, the genome or the proteome or the exposome have to the metabolome? Or if you're a genomic scientist, not considering the influence of how metabolites affect gene expression or gene mutations. So really what I'm arguing and what I argued in the review paper is that we need to think about integrating the metabolome, genome, proteome, and exposome together, and to start considering how to do multi-omics experiments in a more complete or coherent way. So we shouldn't be under or separated as multiple silos. We should be really under one giant silo doing what we call multi-omics. And it shouldn't necessarily be something that is separated, as I showed in the earlier diagrams, but one where you think of these omics fields as being more integrated, more connected. The exposome influences the microbiome influences the proteome influences the metabolome, which then goes and influences the microbiome, which then also affects the metabolome and the genome. All of them are connected in a network. And that network view is something that I think we as a community, in the metabolomics community, but also others in the proteomics, exposomics, genomics, microbiome community also need to think about. So the barriers to multi-omics integration are still considered quite significant. And that's because there are also technological barriers. The instruments that people use in genomics are very different than the instruments and techniques that we use in metabolomics. There is some overlap, I think, with proteomics, but transcriptomics also is a separate field. Exposomics is also drawing in some of the elements of metabolomics, and then the microbiome and metagenomics probably falls more into the sequencing area. We also have barriers where we don't tend to train people in multi-omics. We tend to do a PhD in metabolomics or a PhD in genomics or a PhD in proteomics, but not multi-omics. There's also issues of cultural barriers. People have different ways of thinking about science and thinking about disease in the genomics world. There's also very different perspectives in terms of the role that the environment has in the field of exposomics versus genomics. There's often a view of using too much or too little technology is good or bad in different omics areas. We have challenges with knowledge because these fields do require quite a bit of study and quite a bit of effort. And there are also funding barriers because we tend to silo a lot of our research. For instance, most of my metabolomics research has been supported through a genomics funding agency. And many of the people that review our grants have never heard of metabolomics. We see the examples of where genomic scientists aren't trained in proteomics or metabolomics, proteomics scientists aren't trained in genomics or metabolomics. But if you think about it, and those of you who are now in metabolomics, you actually do have a fair bit of cross-omics training. The metabolome requires that you know something about both the genome and the proteome. Knowledge of these have to be part of your general understanding of both the metabolome and interpreting metabolomics data. We've already seen some presentations where people were drawing out pathways or connecting things where it's not just simply metabolites, but you're also thinking about enzymes and pathways. We also know that the metabolome is significantly affected by the environment. It's affected by the microbiome and by physiology. And this is sort of illustrated in this pyramid where Now, the genome codes for the proteome, which then codes for the metabolome. Yes, they're connected, but the genome really isn't affected by the environment or the microbiome or by physiology. On the other hand, as you climb up the pyramid, the environment does profoundly influence the metabolome. It does profoundly, the microbiome influences the metabolome, and physiology also changes the metabolome. So what you eat or drink changes your metabolome. It doesn't change your genome. Likewise, the organs in your body, they all have identical genomes, but the metabolism in your brain is very different than the metabolism in your heart or in your skin or in your liver. Their metabolomes are completely different. So the metabolome sits at the interface between the genome and the environment. And so it places metabolomics in a really unique position, which makes it, I think, ideal for doing multi-omic studies or understanding multi-omic studies. And this is because essentially metabolites are affected by the genome, they're affected by the proteome, they're affected by the exposome, they're affected by the microbiome. On the other hand, metabolites can also affect the genome. They can also affect the proteome. They can also affect the exposome and other parts of the microbiome. So overall, metabolites really do reflect what we call the phenome, which is in many respects the thing that we're ultimately trying to measure when we're doing biology. So this connection to all of the other omes is something that I think the metabolomics community has inherently been doing, but maybe not recognizing. And it's something that I want to get people to think more about and to think about how their own metabolomic studies can be both intrinsically multi-omic or should be more multi-omic in nature. So if you look at how the trends have been going, as far back as 2003, people started combining metabolomics with proteomics. Then in 2005, they combined metabolomics with transcriptomics. In 2008, metabolomics was being integrated with GWAS studies. That led to what are called MWAS studies. Microbiomics or metagenomics were combined with metabolomics in 2010. The first multi-omic studies where you used more than 2 or 3 studies of different omics disciplines was done in 2011. And then exposomics studies started being combined with metabolomics in 2012. So there's been a trend over the last 20 years as more and more omic studies are being combined with metabolomics. As far back as 2010, there were just 2 multi-omic studies. Today, if you look through PubMed, you'll find more than 500 metabolomics plus other omic studies being reported. So there's a clear trend, and it's not only in terms of combining other omic studies, but it's also the fact that metabolomics is being used as a vehicle towards moving people towards the field of multi-omics. Now, the barriers, as I said, are large, but the way that we can overcome a lot of those barriers have to be done through informatics. And whether it's bioinformatics or cheminformatics, these fields help allow you to both interpret the data and integrate or blend the data. So rather than thinking of the exposome, metabolome, the genome, or proteome as these siloed sections in this pyramid, by blending them, we can create something that is more a description of the phenome, the phenotype, using better software tools. And some of those are starting to emerge now. Now, a lot of my research over the last 2 decades has been trying to create informatics tools to support multi-omics. Most of you are aware of some of the work we've done with the HMDB. That's a human metabolome database, and the database certainly has the word metabolome or metabolomics in it. But in fact, it's a database that's evolved quite a bit since it first came out. There are other databases that we've created for drugs or for yeast or for pathways or toxic exposome or foods or pathways. other tools that we've done, created like Metabolomics to help facilitate this sort of research. These are all databases or servers that we created, as I said, to help make this siloed research a little less siloed and somewhat more integrated. So the Human Metabolome database, I think many of you are aware of. Certainly it's a large database. It has a lot of data. Whether it's about metabolites, their structures, their properties, descriptions, concentrations, reference spectra. There are almost 250,000 compounds, more than 3 million reference spectra, data on diseases. And initially it was intended to be strictly a metabolomics database. But over the years, we've steadily included other omics data sets. So the first release was specifically for metabolomics in 2006. But a year later, we started including DNA sequence data and protein sequence data so that people could search not just for metabolite structures, but also for the genes and proteins that encoded those metabolites. In 2009, we started including SNP data, polymorphism data for GWAS work, and we had DNA search tools. So by 2009, the HMDB was already including metabolomics, proteomics, and genomics data in the GWAS sense. By 2014, we started adding food components and toxic exposure data, bringing in drug information. So now we're starting to include the exposome in the HMDB. And then in 2018, we started adding more information about microbial metabolites and their microbial origins. So by then, we'd already become essentially metabolomics, proteomics, genomics, exposomics, and microbiomics database. The most recent release of HMDB really expanded the multi-omics content even further, and we've been trying to enrich it ever since. So this has been a deliberate plan, and unfortunately, I think people still consider the HMDB as just a metabolome database, but it really is designed to be something more than that. And I think it's arguably one of the most comprehensive multi-omics databases out there. This is another database that was also started off its life as essentially a pure metabolomics pathway database. It was developed to contain just human metabolic pathways that were of interest to metabolomics researchers. So the first version just had about 300 hand-drawn pathways, and those were links to the HMDB and HMDB only. But over the last decade, the small molecule pathway database, or SNP-DB, evolved into something we call the PathBank database. So this again is just this evolution. 2010, it was just a metabolomics database. Version 2 came out in 2014. We doubled the number of pathways, converted a lot of them to become more machine readable, but we also added more extensive protein or uniprot links and more detailed protein metabolite descriptions. And we started including more protein as well as metabolomic pathways. So by 2014, SNP-DB was already becoming a metabolomics/proteomics database. In 2020, we established PathBank and evolved SNP-DB to cover not just humans, but 9 other model organisms. And we ended up going from just 600 pathways to 100,000 pathways. And these included proteomic, metabolomic, genomic, and microbial pathways. We include links to HMDB, uniprot, GenBank IDs, Affymetrix IDs. And so now the database evolved to become much more on multi-omics. PathBank 2.0 will be released later this month. It's expected to have more than 200,000 pathways, and it'll cover same protein, metabolic, genetic, but also have lots of drug pathways, lots of food pathways, a bunch of new pathway analysis features. So again, this is a trend that has been partly pushed by the community, but also pushed by my own team as we realized that in essence, metabolomics is well suited to support multi-omics queries. In 2010, we released a draft version of a database called MarkerDB, and that was initially established to display authoritative biomarker data on metabolites primarily. We wanted to get information about thresholds and sensitivity, specificity, ROC curves for clinically approved markers, and most clinically approved markers are ones that we refer to as the clinical chemistry, things we find from HMDB. So it was a spin-off of the HMDB initially, but over the last 10 years, we started including other biomarkers, not just metabolites and not just approved biomarkers, but also ones that have been described repeatedly in the literature. So again, the evolution of this database went from strictly a metabolomics biomarker database, one which was contained within the HMDB. Then we pulled that data out of the HMDB to make it a little more accessible, and that was done in 2010. Then we started including chromosomal biomarkers in 2016, and then we started including SNP and GWAS type markers in 2019. And then we started including toxic exposome and food biomarkers and food consumption biomarkers. And so by 2021, MarkerDB was becoming just as multi-omic as the HMDB or as PathBank. It's partly because of those efforts of moving largely model organism and human databases that were initially metabolomic databases towards these multi-omic databases that we started on another project about 2 years ago called the Human Microbial Metabolome Database, or MIMDB. The original idea was to have a database of microbially derived metabolites, but we also wanted to include information about the microbes or the microbiome, wanted to include information about their chemistry, also the genes that are associated with them, the effects of food and diet, the compounds and their effect on health. And so intrinsically, this database was designed to be a multi-omic database. It wasn't designed to just be a metabolomic database. So it has thousands of unique metabolites and descriptions about microbial metabolites found in human. It also has a lot of genomic data, almost 2,000 different microbial species with their annotated genome. And then there's links to all kinds of human diseases and mechanisms and associations. So this database is somewhat more complex than something like the HMDB. So users can look at microbes and look at the microbial sequences. People can look at health effects and look at specific links to those. You can look at chemical reactions that are linked to certain enzymes and proteins. And then you can start looking at the pathways and how these microbial metabolites are produced and where they're produced in the body and how they're produced as a consequence of what we eat and their effects that they have on human physiology. So this also gives you a view of the exposome and diet. So this is a new way of, I think, looking at databases, and maybe this is probably the way that the human metabolome database will also evolve. With this sort of multi-omics perspective, linking genes, proteins, metabolites, the microbiome, and the exposome all together. One way of doing this is to create knowledge graphs or knowledge networks, which is an example illustrated here, where you can connect microbes to microbial metabolites to health and disease. And those can then be linked back to specific views in MIMDB database. And knowledge graphs or knowledge networks are, I think, becoming a very useful mechanism to connect omics, multi-omics techniques together. So we feel that the MIMDB is a new kind of metabolomics, multi-omics database. It's still probably in its birthing stages. Yes, there is a focus on metabolomics, but it's really linked to genomic data, microbiome data, food, exposome data, pathway data, health and phenotypic effects. It's already starting to be used to help with the characterization of microbial-derived metabolites, to identify microbial-derived food compounds, and to characterize microbial genomes. It's also being used to determine a lot of the metabolic functions. So all of those things are connected And again, this is building on an advantage that I think the metabolomics community intrinsically has, is that we have to think not just about metabolites, but also the enzymes that make them, the effect these metabolites have on physiology or health, and the effects that it may have on signaling and both protein and genetic pathways. Metaboloanalyst is another tool that I think has also seen a similar evolution, going from sort of strictly, I guess, metabolomics resource to something that's somewhat more. So a lot of you probably use Metaboloanalyst, and it was designed to be a web server to do essentially statistics, multivariate statistics of metabolomics data. And it's gone through multiple releases, starting in 2009, and then later updates in 2015 and 2018 and 2021. And there's another release, I think, that will be coming out later this year or next year. Each of these are designed to make Metaboloanalyst a little easier to use, but also to start including links to other kinds of data or other ways of looking at. So in 2015, we started handling some gene expression data or capacity, and then adding more pathway analysis and more biomarker analysis to Metaboloanalyst. But early on, we also realized that many of the same tools that we were using in Metaboloanalyst could actually be repurposed for other omics applications, because many of the same statistical techniques that are used in transcriptomics and proteomics and genomics are used in metabolomics. So in 2012, we released a tool called Metagenicist for metagenomics and microbiomics. It's not widely used, but it was something that spawned the release of a much more popular version a few years later. We also use the same software framework that was in Metaboloanalyst to create something called INMEX, which is for transcriptomic or combined transcriptomic metabolomic analyses. That came out in 2013. These are just examples of sort of the screenshots of what these tools can do and where you can at least used to be able to access them. INMEX is now folded into another resource. Metagenicist is still around. Anyways, the student I had, Jeff Shaw, went on to do a postdoc and started to do more multi-omics integration. He's now working at McGill University and continues to evolve Metaboloanalyst. But then he's now added the INMEX into the network analyst for transcriptomics. He's extended Metagenicist into something called the microbiome analyst. And he's also developed other tools that are designed to be more multi-omics rather than siloed omics. So MIRNet, MGWAS, OmicsNet, Omics Analyst. And then to help support these, he's also created a number of databases. But this is an example of how, essentially taking his PhD thesis work, which was on Metaboloanalyst, he was able to take existing tools and just sort of rebrand them. In some cases add a few more tweaks and bells, but not much really, to create a whole library of very useful tools. So in that regard, I think it highlights the fact that what you do in metabolomics opens the door to you to do lots of other multi-omics analyses. And the path to do that is often through software and databases. So I think what Metabolomics has done is it's sort of helped take a lead in multi-omics integration, whether it's through the tools that I've highlighted with HMDB or PathBank or Metaboloanalyst or MindDB. And this is from things like software, it's things like databases, it's a variety of analytical tools that have been developed. And it's also, I think, led to a number of metabolomics cores to start to offer not just metabolomics support services, but also proteomics support, and increasingly now even gene expression support. So our own Metabolomics Innovation Center, or TMIC, is starting to offer both proteomics and metabolomics and exposomics, and is also starting to explore microbiomics and other things as part of that multi-omics approach. The other thing that's happening is that integrated multi-omics analyses are starting to appear in a variety of precision health or precision medicine or precision wellness companies. And I think this is, an important thing to remember, is that this isn't just for the sake of saying, look how many balls I can juggle or how many different methods I can use, but the perspective of having a broad collection of information makes understanding of diseases and the characterization or prediction of diseases more accurate. And so I guess you could call it holistic omics or holistic wellness. But when they've started using multiple omics techniques, these companies have been able to report some pretty impressive improvements in human health, or have been able to uncover a number of hidden disorders. So these are examples of some of the companies that have started appearing over the last decade. There's Molecular U, which is based in Canada. There's Savaris, which is based in the States. Human Longevity Incorporated. Atlas, which is in the UK. Viome, which is in the States. YOLO, which is in the US. Day 2, which is in Israel. Zoe, which I think is in the UK. All of these combine the metabolome with the microbiome, the genome with the microbiome, genome, phenome, metabolome together. And they all, either through measurements of a blood sample, a urine sample, a fecal sample, provide some suggested improvements or alerts about your health and what you can do to fix things. This is data from Molecular U, where they used a combination of both proteomics and metabolomics to first characterize a person's general health score, and then to put them onto a 3-month action plan where they had to improve or modify their diets, their activity, and then how they performed 3 months later in terms of their overall health score. So everyone basically saw fairly significant improvements in a variety of health areas. And a lot of those things actually, the determination of the health index was made from using reference values in these public databases from the MarkerDB, HMDB, and other tools that have been made public. So this is something that is now quite feasible, and where multi-omics is being part of a practice of medicine, and where it's not just companies, but now more and more what are called concierge medical clinics are doing the same sort of thing of integrating multi-omics to make assessments and to implement action plans. Now, as someone who maintains a lot of databases and web servers, there's still a lot of challenges ahead for us, both in maintaining and updating the omics data sets. One thing we've noticed over the last 10 years is the volume of data, the number of papers being produced is actually just too much for our own team, our database curators and our bioinformatics team to maintain. And so what we're having to do, along with people like Jeff Shaw and others in the bioinformatics world, is starting to move to things like natural language processing, chatbots, large language models to do the things that our curators can no longer do, to create connections, to create knowledge graphs, and to introduce something called chatalytics or chatbot analytics. These are still evolving, but over the next year or two, I think a lot of these things will become part of what you'll see in both HMDB and MetaboAnalyst and other databases. And these will use a variety of what are called fine-tuned large language models to do fact extraction. We'll use natural language processing techniques to get ontology triples. They'll build large knowledge networks from the literature data. And from those knowledge networks and the large language models, they'll be able to assemble, I think, very useful descriptive tools, some which will auto-update the databases, but also allow people to make very simple chat-based queries. So I think to wrap up, I want to emphasize that Metabolomics really is and has become the ideal vehicle for integrating other omics techniques together to really do multi-omics. I think we've already started to see an explosive growth in the number of metabolomics plus other omics studies that are already being done. And I think I've shown you how existing software, existing databases are being repurposed by members of the metabolomics community to make them more multi-omics in nature and make them more accessible. I think we're also starting to see how generative AI and generative machine learning are also going to be used to help make multi-omics studies much easier to do, much easier to analyze. And I'd actually urge you as young scientists to consider how you can use other omics methods in your own metabolomic studies to make things, I think, more comprehensive and more interpretable. So with that, I just want to thank members of my team, people who've worked with us over the years to contribute the software and the databases and our funding sources. And thank you for listening.
Thank you so much, David. Thank you for this really impressive work accomplished by your team. And now I see that there is a question for you in our chat. For instance, you have a question from Jean-Charles Martin that asks how long you think it will take to implement the efficient AI tools for large multi-omics data interpretation, because it seems to be the key.
Yeah, and I agree that a lot of the multi-omics tools do tend to focus on fancy graphs that kind of look like hairballs in the end. I think that process is going to take another year. And it'll probably evolve over the coming years. So there is pretty active effort by a number of groups to use the AI tools and Chatlytics to make things more interpretable and to produce more essentially textual descriptions of how to interpret what's being produced or what data is being generated. I think it's also going to be very much guided by users and what people like and what they don't like, and I guess examples of scenario building. We're certainly anxious to get people's opinions as some of these things start rolling out. And as I say, we are actively working on this, and I think it should happen within the next 12 months.
Cool. So Lina ask you, so thank you for your presentation, and ask you when you use this multi-omics approach, is it based on qualitative or quantitative data, or maybe both?
So my own personal preference, at least with multi-omics data, is to try and work with quantitative data. And the examples I was showing about how the precision medicine or precision health and wellness companies are doing things, they use fully quantitative data. So that integration, when you have quantitative data, is much, much easier. Now, it is possible to do multi-omics integration with a mix of qualitative and quantitative data, but the interpretation isn't as powerful. One of the reasons why I like quantitative data is that biomarkers have to be quantitative, and biomarkers are linked to disease diagnosis, prognosis, and interpretation. So most of our interpretation for multi-omics is limited to quantitative data, and I think that's something that the community really needs to think about and address.
Okay, thank you. And when you are working with data from different analytical techniques, you are working on pre-processing and harmonization, and the issue also arises between sample types. Sorry, maybe in the context of multi-omic, how can we overcome this problem and be sure that all the data has the same repository somewhere and that we don't observe any bias? as you said at the start of your presentation, there are technological barriers and then there are scale barriers. Even if we have AI, certain standard conventions need to be defined. So how can we remedy this? Maybe you can read.
That's a big question. But I think it all goes back to this idea of moving more and more towards quantitative omics, whether it's quantitative proteomics, quantitative metabolomics, quantitative exposomics, quantitative microbiomics, whatever. They are all possible. The techniques are there, the standards are there, approaches have been established. I think there's been, some resistance within the metabolomics community, some resistance also within the proteomics community. But I think people need to realize that the only way that markers or metabolites that you measure, proteins that you measure, the only way they can ever be translated into some sort of clinical practice is they ultimately have to be quantified. They won't be approved by the European Health Agency, they won't be approved by the FDA, they won't be approved by Health Canada to become useful markers. When you quantify things, 1.2 millimolar means the same in Canada as it does in France, as it does in the UK. It helps standardize things. It harmonizes things. There are many examples of people conducting quantitative omic studies in multiple different labs and getting essentially the same results. However, when people try to do untargeted studies, the results have been utter disasters with absolutely no reproducibility. So I think I strongly recommend people to just look at some of these ring trial studies where quantitative efforts were undertaken, and all these issues of pre-processing and harmonization and everything else just go away. So it goes back to the need to really focus on quantitation.
Okay, good. And maybe a question about possibility to connect the MIME database to MicroBlast tool developed in the team of Peter Dorostein. Maybe, probably you know that new tools that allow to make the blast on spectral data.
Yeah, I think we are working with Peter as part of this larger NIH project. And so hopefully we'll be able to make some of those connections over the coming few months. But MindDB did grow out of the same funded project with Peter. So I think it's a good suggestion, and it's certainly something that we're hoping to work on in the next few months to do.
Okay, so I do not see more questions. Maybe Loic, you can make the final conclusion of this webinar.
Yeah, so thanks again, David, for having accepted to share with us your thought about metabolomics and multi-omics. It was a great talk, and now I have a few advertisements to do for the next webinar and what happens next in the French Metabolomics Society. So I'm just gonna share my screen to share with you only 2 slides. So first thing first, wait. So yeah, we'll be hosting the next webinar on the 7th of December. The keynote speaker will be Yann Guiton, and this webinar will be mostly about annotation and how we can improve our annotation of LC-MS data. So stay tuned, and we will send the full list of speakers in the very near future. The second thing is about the École Automnale of RFMF, the Autumn School of RFMF, which will be only in French. for all adherents or the RFMF that are under 33 years old. It will be in Lyon in France, close to Lyon, and it's limited to 35 participants. So if you haven't registered yet, do it quick because there are a few spots left, but not many. So feel free to register, and I'll just use this information in the chat before we leave.
So thank you, Loic.
Yeah, no worries.
Thank you all. Sorry.
No, not a problem. I'm just copying the info in the chat so you can remember. And thank you all for attending. And see you next time. I'll let you the mic.
Bye bye, everyone.
Goodbye. Thank you.